Anglian have been asked lots and lots of questions about hypochlorous acid over the last few months, questions we didn't know the answers to, so we set about trying to find out the answers to the questions. So we did a bit of a deep dive to find out what is it safe, how can I use it, what shouldn't I do with it, that type of thing. So if we move on to the next slide, please. So I'd like to introduce Dr. Lack and Professor Reynolds, along with my colleagues Murray and Fraser. Now, Professor Reynolds is, in my opinion, the world leading authority on hypochlorous acid. If there's something that Darren doesn't know about it, that's Professor Reynolds. Nobody does. So he's very brilliant. He's joined us tonight, and we'll be hearing a lot from Professor Reynolds. We've got Dr. Lack, who many of you would know. He's been very involved in the recent FGDP guidelines, including specific interest in research onto the fallow period after the AGP. So next slide, please. So hypochlorous acid, what is it? We're treating this a bit of a boot camp in some ways, that all of you would know what hypochlorous acid is, and indeed probably using it. But what you may not know is the number of derivatives of hypochlorous acid. It's incredibly important to use the correct derivative or the correct makeup criteria for the purpose of which you're using it for. So what is it? In the simplest form of the term, it is electrolyzed salt water. Hypochlorous acid occurs naturally in our bodies. It's one of the only known agents that's both non-toxic to delicate cells while being lethal to almost all known bacteria and viruses. So one question you could ask is, why hasn't it been used extensively for many years? I understand that Michael Faraday invented it 100 years ago. Sort of where's it been? You'll find out the answer to that question later. Next slide, please. So not all hypochlorous acid solutions are the same. Terms to look out for and to learn more about, you can see them on the screen here. That's ORP, oxidative radiation potential. ppm, that's the available free available chlorine measured in ppm. And the pH, which is the potential or power of hydrogen, or your pH scale, as you would know it more commonly known as. So the various applications of decontamination of hypochlorous acid is decontamination of surface, decontamination of surfaces by air, and decontamination of air. So that's me done for now. I'd like to please hand over to Professor Reynolds, Dr. Lack, who's going to take us through in respect of dentistry and his involvement in it. Thank you, Dr. Lack.
Thank you, Phil. First of all, thank you to you and Anglian Dental for inviting me to be part of tonight's webinar. I have to start off by saying that actually I am no expert at all in HOCl or its fogging applications. I'm here simply because I became very involved in our return to work guidelines as a representative of the Alpha Omega Dental Fraternity Scientific Committee. And we were invited to sit on the FGDP Future Planning Task Group to create guidelines for us all to return to work. So I do have some insights into the guidelines, how they were put together, how they were meant to be followed, and maybe even how they're meant to be interpreted, but specifically why most guidance documents do not mention fogging as a possible method to justifiably reduce our fallow air times. The BDA mentions it really more so as a surface cleaner, I believe, but not actually as a fogging application. And of course, for all of us, these fallow air times are a major issue. They really don't allow us to provide enough patient care in a timely manner. And they also don't allow us to really practice financially viably, which can't be ignored at all. So we need to try and find some method perhaps of helping to reduce these fallow air times. Before we sort of dive into it though, I do think it's really important that we're all on the same page and we have a really good understanding of what the problem is and what we know about it so far. the big deal in dentistry is that all of our patients come into the room and they breathe, which is good, and they might sneeze, they might cough, and they speak. And in doing all of these things, they may create an aerosol of some sort. But in dentistry, we tend to ramp that up a little bit when we fire up a high-speed handpiece or an ultrasonic tip or a three-way syringe in their mouth, and we create a bigger aerosol, and we mix that aerosol with the saliva in their mouths. And we create these sort of droplets, and the droplets can either be large or small. The large droplets we tend to classify somewhere around 5 to 10 microns, all the way up to 50 microns. And the beauty of large droplets is that they tend to fall very close to the chair. They don't tend to stay floating in the air. So they may fall within 1 or 2 meters of the chair. And we've been dealing with these throughout all of our careers, really. And certainly our level 2 PPE procedures that we have in place now, including your plastic apron and some eye protection, a visor, surgical mask and gloves, are all very effective at dealing with droplet precautions. But the real problem are the other particles, the small particles that might be 5 microns or less. And although all particles can potentially have virus on them if you're dealing with an infected patient, the concern is that the 5 micron particles might actually lodge lower down in our lungs, and that is more likely to cause a worse case of SARS-CoV-2. The lower it gets in our lungs, the worse we tend to see the illness become. So that's really what our concern is that we may create this aerosol with small aerosols that will remain floating in the air, and we'll breathe them in. Either we will breathe them in, our staff will breathe them in, or the next patient will breathe them in. And so I would like to point out that to date, we do have some research on these aerosols and their ability to survive in the air. But I would like to point out and really emphasize that the research that we have is experimental. And what they did in this research is they took virus particles and they created an aerosol with it, and they shoved it into this chamber that spins and keeps the aerosols floating for extended periods of time, keeps the virus up in the air. And what they found in one study up to 3 hours and in another study up to 16 hours is that the virus that they were then able to recover appeared to be viable virus. It appeared to be able to infect us. So looking at that research, it certainly is possible that there is virus in the air when we create a dental aerosol. But what we don't know yet is whether specifically dentally created aerosols, not experimentally created ones that they filled with virus, but dentally created aerosols even have the virus. In all likelihood, because the research looking at saliva certainly shows us there's plenty of virus in saliva, in all likelihood, the aerosols that we generate will have some virus if you're dealing with an infected patient. But we also don't know whether the amount of virus that's in that air will ultimately be able to infect us or the next patient. So even though there are still some very big missing aspects of the puzzle, we still have to consider that it's plausible that there will be virus floating in the air for an extended period of time that is viable after an aerosol generating procedure. And therefore, we need to make sure that we're cleaning our air environment after these procedures are done. So, as I started off by saying, I'm not an expert in HOCl. Professor Reynolds is. But what's become obvious in going through this process of researching fallow air times and working with the FGDP on their guidance document is that there do not appear to be any real experts in fallow air time at all. In fact, when we were going through the process of researching the FGDP document, we repeatedly asked Public Health England for evidence of the need for a 60-minute fallow air time, of which they could not provide. They pushed it up to NERVTAG, which is the New and Emerging Respiratory Virus Threats Advisory Group, and they also did not offer any proof of the need for a 60-minute fallow air time or why they focused on 60 minutes as being the key figure. Really, the closest thing we have to research on this topic is what I would call more the splatter and droplet research groups. who have looked at aerosols with dye in the aerosols that they've sprayed into the air with high-speed handpieces or ultrasonic tips, and then they'd look to see where they can see the spray around the surgery. Or they might have placed agar plates around the surgery to see what bacteria would land on these agar plates and grow over a certain amount of time and at what distance from the chair. But in terms of true research, looking at virus particles in the air, we really don't have anything to hang our hat on. And indeed, even as we speak tonight, we just read on the BDA website that Public Health England has undertaken a bit of research into this, and we may even get an announcement tomorrow on our fallow air times. And the Scottish group, the SDCEP, is also currently conducting a rapid review with the Cochrane group. And they're looking at both fallow air times as well as all of our mitigating elements, such as rubber dams or high volume aspiration. And hopefully they'll report on that very soon as well. But for now, we really are stuck. We are not able to see as many patients as we need to see. And we definitely need to find a way to get back to viable, more reasonable appointment times and fallow air times. And it's possible that HOCl fogging is the answer. HOCl and its derivatives has been around for a long time, as Phil said, and it's very well proven in the food industry and water disinfection industries. And Darren, as we mentioned, has been involved in this research for many, many years, and we'll talk about that in a minute. There's very good evidence for its ability to kill microbes on surfaces when used as a spray from a spray bottle. But to date, there is no proof of its efficacy specifically killing viruses in the air. And that's really why, in a nutshell, most guidance documents don't talk about HOCl fogging as a possible justifiable method to reduce our fallow air times. It's not because it doesn't work, and really, on the contrary, it has fantastic promise for use both in dental surgeries, in any healthcare environments, and really indeed in any indoor spaces where there might be a threat of an airborne virus or any microbes for that matter. But it's not yet proven. None of the applications are proven. And I really applaud Anglian Dental for putting on tonight's webinar and being so responsible in putting it on, because the more they researched it, the more I have researched it, and certainly the more we spoke to Darren, who knows more about it than any of us, we have found that there's quite a bit of misinformation out there. And so tonight is equally about learning about fogging, but also a little bit of myth busting. And also really, I suppose, a take-home message to be very, very cautious that if you are currently fogging and you are using the fogging liquid and fogging machine that is currently available to justifiably reduce your fallow air times, to our knowledge, there is no machine yet that is doing that well enough for you to hang your hat on it and say, I now only need, say, a 5-minute or a 10-minute fallow air time. So be very, very cautious if you're going to rely on that as your justifiable method to significantly reduce fallow air times, because again, to our knowledge, that product and that combination of product and machine and parameters doesn't exist yet. And certainly the research proving its efficacy is not out there yet. And I stress yet. So that's all I really wanted to say for now. I'll hand back to you, Phil, and to Darren.
Thank you, Dr. Lack. Great to get it straight from the dentist's perspective. Hear it firsthand, feel it really firsthand. So thank you. We'll just hand it back on to Professor Reynolds to help some of the myth busting. Thank you, Darren.
Hello, everybody. Nice to see you all. Yeah, I echo Jonathan's thoughts really about thanking Anglian Dental for providing the opportunity for me to sort of speak with a sector I don't normally deal with, if I'm really honest about it. But I've been involved in looking at decontaminants, including hypochlorous acid now, for probably over 25 years or so. We've used it in other sectors, in the food sector in particular, and also in the water sector. So we've been using it for decontaminating microbiology or pathogens on food, and obviously disinfecting water that we treat in off-grid situations. We've also done a bit of work on looking at the role of hypochlorous acid or electrochemically activated solutions within the healthcare space as well. And it's a powerful oxidant. It has a lot of really interesting properties. It's principally driven by chlorine, so it's got chlorine in the chemical form. It's HOCl. It's neutral. It's not charged, and that's actually quite an interesting property that it has, because it allows it to penetrate and get access to the internal machinery of bacteria, and actually also viruses too. And it's able to move unhindered through the membrane wall often, or through an envelope, and then oxidize some key components of those biological entities. So it can oxidize, for instance, proteins on the surface, receptors on the surface. It could maybe interfere with the genome on the inside. And that sort of mechanism of kill is not too dissimilar to what swimming pools use. So I do a lot of work with people in swimming pools, and they use chlorine chemistry to provide effective disinfection within swimming pools. And you can make hypochlorous acid in a number of ways, and I think we'll be talking about that a little bit later on. But you can basically add chlorine gas to water. That's a little bit tricky. You can also use hypochlorite. So most of you will be familiar with bleach. Actually, hypochlorous acid is part of that chlorine chemistry in producing hypochlorite, and one of the key elements of shifting the chemistry into a place where you can see the formation of hypochlorous acid is about a pH 5. So the maximum sort of amount of hypochlorous acid in that chlorine chemistry exists at around about pH 5, pH 5.5. These days, a lot of technology is electrochemical technologies. They're like batteries. They're in place, and they're very good at producing hypochlorous acid. But the things that you've really got to look out for are things like the pH. So the pH is very important. So pH 5 is a good indicator that your solution, if it is indeed derived from an electrochemical solution, is indeed hypochlorous acid. Parts per million refers really to the active amounts of chlorine that you have. And again, this is, you can get solutions that are as low as 40 to 50 ppm, that are indeed as high as 200, even 500, even 1,000 ppm, depending on what the purpose is of the solution that you're dealing with. And of course, the ORP is a very interesting and important parameter. It's not a measure of chlorine, and chlorine's not straightforward to measure, but it does give you an indication of the oxidizing potential of the solution that you've produced. And again, that ORP, the oxidation reduction potential, really is a dimension of oxidative or oxidation ability. So if you think about it, you're putting out your hypochlorous acid. It's meeting a challenge, hopefully a biological challenge, and the hypochlorous acid is being reduced. The ORP can give you some indication as to the efficacy of the starting solution. And in fact, we often use ORP to measure the process of change. So as that hypochlorous acid is reacting, sometimes we can glean something from the ORP. So it's very good quality indicator, for example, if you've stored those things. Perhaps the biggest advantage over recent time has been the ability to produce hypochlorous solutions in stabilized forms, because they've been a little bit tricky to do that, particularly through electrochemical generation, which is actually where a lot of the interest lies, because you can produce that on demand. Okay, so I'm not quite sure what you want me to quote CDA, but I can certainly talk about the EN test 14476, so maybe Phil can come in and help me out on the quote. But the EN test, all disinfectants and all biocides are really measured against normalized testing regimes that are in place across Europe, certainly in the UK, and often in many parts of the world. And these are designated as European normal testing regimens. EN 14476 actually refers to the efficacy of a particular disinfectant in suspension and its effectiveness against viruses. So often it's a very complicated landscape. You will have lots of different EN numbers, but the basic ones to look out for are the bactericidal one, which I think is the EN 1273, and the other one is the EN 14476. And the 14476 denotes the effectiveness of a particular biocide or disinfectant against a viral challenge. And of course, this indeed is what we're looking for at the moment to combat the spread and transmission of COVID-19. And these EN tests really help to inform the qualities of a disinfectant that you're using. And you've really got to sort of really understand that landscape to ascertain whether or not your disinfectant is best applied after a clean or during or before a clean, if you see what I mean, because some disinfectants are much more effective when they're applied in clean conditions, we use that term microbiologically, than they are when they're applied to a challenge that has got other substances in there other than the bacteria that you're looking to kill or to inactivate. And the performance of those things really depend on how you quantify the efficacy, and the EN testing landscape is very important for that process. Hypochlorous acid is used in many parts of the world. It's a very well-known biocide. It's not very well known here in the UK, and in fact in a lot of other places in the EU. That is a function, I think, of the legislation and the biocide product register. It's a function as well, I think, of the market. So it's kind of a market issue as well, as much as anything. But its efficacy is definitely proven on surfaces and in suspension. That is not really in question. And its application is definitely well proven in terms of spray. So Jonathan was mentioning, spraying from a bottle and maybe spraying on dirty surfaces, etc. Those things are very well understood. What is not well understood is its performance in as a aerosolized entity. So how does it perform in that respect? There is another EN test which has just recently been adopted, which looks at the effect on a biological challenge on a wall, actually several biological challenges in a room, and you're looking to achieve something like a 4-log reduction of those challenges after a sustained period of fogging. The trouble with that is what it takes to reduce a bacterial load on the surface, particularly a surface that's impacted with other things like biological material, is very different to how the efficacy of a decontaminant like hypochlorous acid would indeed perform in the aerosolized dimension. And what is very sparse at the moment is looking at the effect of aerosolized antimicrobial particles on biological pathogens in that 3-dimensional spatial entity. And that is where the challenge lies here. You can make your own. You can do that by getting a really good chemistry book or just Googling it. I wouldn't recommend that. You can actually buy tablets as well. There is a chemical formulation of that, but you really need to understand some of those other parameters, such as pH, a little bit of molarity and a bit of chemistry, all those kind of things, in order to be confident that what you're producing at the end is, number one, at an appropriate concentration for the job that you require. Number two, that it's stabilized and has a reasonable ORP value, and also, of course, has a pH in the range of 5.5. The good news is technology is really moving on. So the ability to produce HOCl on demand in a space when you need it is actually is there already. So the technology exists. What doesn't exist is just some of those knowledge gaps, which actually is what we're looking to do at the moment. Okay, so again, how you deploy the decontaminant is really, really important. So again, if you spray something, it's going to, you know, the mechanism of the decontaminant going through that delivery system could well have an impact. If you're going to fog it, then the size of droplets that you use or produce is going to have an impact. The mechanism of aerosolization, whether it's transducers or whether it's a mechanical whirly device, is also going to have an effect on the overall efficacy. And the trick in all of this is to understand which delivery system is the most effective for the job. And I always see it really as it's just a target and shoot mentality. What we've got to do is deliver the decontaminant, in this case hypochlorous acid, to the target. And our target here is aerosolized viral loads. And that is really where we need to do a little bit more work in order to really understand what we're doing, and more importantly, show and demonstrate that what we're doing is actually being effective. There is other well-known decontaminants. Ozone is one of them. Hydrogen peroxide is another. And they're very, you know, they are used. Ozone's used a lot in the food industry. And hydrogen peroxide is used a lot in deep clean services. But those services understand very well some of the occupation exposure limits that are associated with those types of decontaminants. They are both extremely strong oxidizers. Their mechanisms are very different. They're driven by oxygen, really, as an oxidant as opposed to chlorine. So their mechanisms are different. The time and kinetics involved are very different. So in my feelings are that in this situation, given that you have people, given that you have spaces which need a fast turnaround, that those approaches are not appropriate for what you need. What you need is a disinfectant or a decontaminant that really displays two very clear properties. Number one, it has to be biocompatible and safe. And number two, it has to be effective. And those two things have to be firmly implanted, I think, in all of our thoughts when we're thinking about tackling this particular issue. So that is the biocompatibility and the effectiveness. And in this situation, I don't think hydrogen peroxide or indeed ozone or anything like that, it really displays both of those qualities. They certainly don't display the qualities of biocompatibility. What we really need to do is I'm afraid is really fill in the blanks using science to really show and demonstrate that these types of processes that we want to deploy, aerosolized HOCl, is effective for cleaning or decontaminating air. Now, the good thing about air is it's usually a very clean medium. Even when you have aerosolized generating procedures that are filling the room with viral loads, really on the big scheme of things, we're looking at a clean medium. That's good news, because that means the efficiency and the effectiveness of our decontaminant is enhanced. Because if it's in dirty conditions, we know that the quality of that disinfection is reduced. So that's a really good start for us. What we need to do is to really understand what role and how important the particle size is. And we think at the moment, the particle size we should be looking to deliver is about 5 microns. So a lot of foggers and sprays do not really display that level of control over the aerosolized particles themselves. And it's always a distribution. And what you're looking for is the vast majority of aerosols to be produced being 5 microns in size. Now, your COVID-19, or the virus that gives rise to COVID-19, SARS-CoV-2, is 100 nanometers in size. So to put that in perspective, 100 nanometers is about 1,000 times thinner than a hair on your head. So these viral particles are really small. Reality is they'll probably be you know, they will be together, bunched together, probably as part of human fluids that have been expelled, or maybe even because of static forces, they'll be forced together and come together and sort of, you know, form much bigger aerosols. But the role that the particles sizes of the decontaminants have on those aerosolized viral particles is not that well understood, and that's a key knowledge gap. Safety considerations for humans and environment. Well, again, I think the reason why there's room for optimism is because in the literature, the effect on materials is very negligible. And actually, in terms of toxicity, the effects or the toxicological effects appear to be very low. So there's been some work, quite a bit of work done on looking at hypochlorous acid as a topical wound agent. And as part of those studies, they've done a lot of work on its cytotoxicity and its genotoxicity. And there's even been some limited number of animal studies. And to date, all of those studies that have taken place have shown that in comparison to all of the other decontaminants, hypochlorous acid is very, very safe. And in fact, our bodies are very used to hypochlorous acid. It is something that we produce in our bodies as part of our immune system. And it's a natural defense that we have, the production of reactive oxygen species within our immune system to combat alien invaders. So the news on that front is good. What is less understood is the effect that aerosolized hypochlorous acid could have on the lungs or the respiratory tract. And that information, to my knowledge, is not there yet. We have a plan to do that. We have the possibility of doing that, but that information and knowledge is incomplete.
Excellent. Thanks, Darren. That was very insightful, very interesting. Moving on now, a bit more to apply what we've just heard about. We're delighted to introduce BioFog, which is a platform we've set up, Anglian Dental set up, which will be a source of information and updates as things develop. And we've heard about a consortium group that's testing this in a lot more detail, and we'll be posting updates on the BioFog website. We'll also be introducing a fogging machine, and we'll hear more later about that, and also a fogging solution which has been especially developed for use to fog in dental surgeries. So keep an eye on BioFog. Moving on to the next slide, fogging in a dental surgery, a lot more complex than we initially thought. It's one thing we do know, it's certainly not the case of just buying any fogger, any hypochlorous acid, turning it on for any period of time. That is really not the correct approach. As Dr. Lack said, there's a lot we still need to find out. We need evidence, we need results from labs, we need to learn more about the parameters that are important to ensure its efficacy. Some of the key considerations for fogging in a dental surgery, if we can have the next slide. The process and the method is really quite important. Sorry, just the previous slide there. The process and the method is really quite important. If you can just go back one, thank you. The fog quantity, how much fog is put into a surgery. Surgeries are different sizes. There needs to be some method around working out how much fog we put into a surgery to clean it, and the parameters we need to measure are all key things we need to find out more about and learn more from. Some of the parameters we've been looking at: humidity and dew point, the temperature of the environment. It's all very interesting, but until we have more evidence, we're limited in how much we can say right at this stage, but there's certainly huge potential. Clearing the fog after you've fogged a room, the humidity rises significantly, and that can be uncomfortable. So bringing that back down is something, is a challenge. We've done tests using air purifiers, dehumidifiers, and there's ongoing research into the best way to keep that under control. The next slide, we're discussing the fogging devices themselves. Awful lot of time and effort has gone into our research so far on this. We brought several machines, taken machines apart, tried to understand how they all work. We've actually got a machine in development. It's one you can see on the on the slide deck there on the left hand side, which is that's at the moment that's like a prototype. We have a patent pending for some of the technicalities of that machine. And what's important are a few key metrics here. It's got to be a powerful machine. It's got to get fog into the room quickly. It needs to be portable. You need to be able to move it between surgeries. And the tank material where it stores the hypochlorous acid is important. HOCl has oxidative properties, so what that's made of is absolutely essential to ensure it doesn't corrode. And then having a sensor so you can tell what the humidity or parameters inside the room. You can read what that is from outside the room. You can adjust the machine. It needs to be easy to use. And lastly, and probably most importantly, as being covered off already, is the particle size. What we need is an ultrasonic humidifier, and that's critical to ensure the particle size is small and effective. The next slide here will show us just a very basic illustration, really, of how, and I hope explain why the particle size is so key. That can just be played. So what this essentially shows in the dental surgery, that with a handheld device like a sprayer, you can't prescribe the fog amount scientifically. Large particles are emitted. They have a low surface area, and there's room for human error with a handheld device. It takes longer. Someone has to be in the surgery, and with a larger surface area, a smaller surface area through large particle size, the chance of it being effective against particles in the air is limited. What is important, as we've just said, is if we have a ultrasonic humidifier, it emits a much finer mist. It's a dry mist. It's not wet like you get with a large particle, so it wouldn't leave a wet residue on any equipment items. No need to dry up. It wouldn't be wet to touch, and it's much quicker. It can be done. There's a quicker kill time because it's a larger surface area, and it can be done just by leaving a machine in a surgery, and it's much more effective in its ability to kill with a small particle size. So I think that's really the message of that brief illustration there. The next slide is a demonstration video which we shot in a dental surgery. What this is showing, just before we hit play, it's we used our prototype in a dental surgery. We used an ATP tester, and we swabbed a surface of an area before fogging. We then ran the fogging machine, and then we measured the ATP test afterwards. This, understandably, is measuring the contaminant on a surface, but by decontaminating the air, we are working on the assumption that it was it was decontaminating the air around the surfaces. We ran the machine for 2 minutes. The room humidity rose by approximately 20%. So that was an interesting test we did, interesting video there. It shows the potential that hypochlorous acid has. We completely agree this isn't particularly scientific, perhaps a rather crude test, but it does give some idea as to the potential it has, and we're excited about the research which is being undertaken to give us more information around the details on that. Next up, we're talking about the effect on hypochlorous acid on dental equipment. And this is a question, being dental engineers, we get asked a lot at the moment. And what we did to experiment more with this is we took some components of a dental chair. We took an upholstery swatch, we took a circuit board, and we took some painted components, and we soaked them in hypochlorous acid for 4 days. Understandably, if a surgery is being misted, it would have a much lesser effect on components. By soaking it, we hope that is a fairly rigorous test on the components, And what we've got here, we can see an upholstery swatch in the left-hand side. This was interesting. After 4 days of soaking in the solution, it showed no discolouring at all. The sheen was still on the upholstery, and the hypochlorous acid hadn't discoloured at all. The screw, the one on the left-hand side, is one that was soaked in hypochlorous acid. Again, the paintwork was all intact. It hadn't rusted in any way, and it was great to see that it hadn't had significant impact on it at all. The circuit board we submerged is from a spittoon valve, and although you can see oxidation on the surface of the control board, the circuit board still worked perfectly well on testing afterwards. It has 24 volts running through it, and so that was excellent to see that. One point to note is, in speaking to manufacturers, we've spoken to the main manufacturers of dental equipment in the UK. Manufacturers are taking a very cautious line on this, understandably. They're not recommending it is used. Perhaps there are other motives for that. Some of them have their own product lines they want to sell for cleaning products, so they are not recommending it. And there obviously is the chance that warranties could be invalidated if you're not using their specified solutions. But the potential it has is very exciting, and this simple test, and it really did show that its potentials in cleaning and misting dental equipment, fogging dental surgeries, providing it's turned off and circuitry isn't live, could be completely safe. Next up, I'll hand over to Fraser, who will talk more about a solution developed for hypochlorous acid fogging.
Thanks, Murray, and that was very insightful. I hope everyone's found what we've just covered off there very useful, and hopefully it's answered some of the questions that there are. We've tried to answer some of the stuff that was been coming in over the past weeks and months. So H++ is a unique stabilised hypochlorous acid solution that's been developed over many months and is perfecting the recipe. It's made in the UK. It complies with the standards that we've mentioned earlier, the EN 1276 European norm standard for bacteria and the EN 14476 virucidal European norm standard. It's developed specifically for fogging, so air decontamination. So it has the nice high ORP that's needed. We touched on that earlier. So what that does mean is it's also extremely good as a surface disinfectant if you wanted to use it for that, with a very rapid kill, proven to kill bacteria in under 2 seconds. Can be used decanted into a spray bottle similar to the one shown on there with the continuous spray pump action. More on that to follow. And it will be available to purchase very shortly from the biofog.co.uk website that Murray touched on earlier. So please do watch out for that. One of the important things in how it might differ from other products that are on the market is its 12-month shelf life. And also, as the next slide shows, it's very minimal residue. Now, we haven't really covered this off particularly, but hypochlorous acid, if it's left to evaporate and not wiped off a surface properly, can leave a residue. And the H++ stabilised solution we found in our tests on Petri dishes to leave 90% or more less residue than the other common products such as Optazil or Nemesis and other well-known brands that we tested it against. So it's just an important point to realise on the residue side of things. So please do watch out for that product coming live, available to purchase very shortly. biofog.co.uk is the platform. Next, we'll move on to making your own hypochlorous acid. We touched a bit on this already earlier, and you might say, well, why would you make your own? You can buy a H++ in a bottle, excellent solution. Yeah, and that is great for the fogging process and rapid surface cleaning. But with the Toucan Eco range that we're showing on the screen here, you can make a grade of hypochlorous acid that, yes, is not suitable for fogging and air disinfectant, but is very good for surface cleaning and can be used as a general disinfectant and all-purpose cleaner and as a deodorizer. Very simple to make using these machines. All you need is water, salt and electricity. Very environmentally friendly way of cleaning your practice with with these machines, you don't have to. It basically strips out 80% of your current chemical spend. So all those bottles of cleaners that you're buying at the moment, you're saving all those plastics, saving the environment, and using one of these machines to make the solution. So you're never going to run out. All you need is your table salt and your water. Plug the machine in. The one on the right is the Toucan Eco 3, so for smaller quantities, a litre of solution. It also comes with the spray, continuous spray pump action bottle there for actually applying it to the surfaces. Then on the left hand side you've got the Toucan Eco Active, which is for larger quantities of the solution, can be plumbed in to the mains or filled up with jugs, and makes a larger quantity of solution, so 10 litres in around 15 minutes. In terms of the capital cost of buying these, it's been proven to be paid back within under 12 months, so a very quick payback time there, and very minimal ongoing running costs. So just your salt and your water, electricity, about 1/10 of the amount of what it takes to boil a kettle, if that helps put it into perspective. So lots more information on this is on our website. They're available to purchase on our website. There's downloads on user guides and operation and installation manuals on our website. So please do check that out for more information. And I'd like to just wrap up that section now and thank everyone for listening, and hand over to Phil for some questions and answers, which is what we've all been waiting for. Over to you, Phil.
Thank you, Fraser. Thank you, everybody, for taking part. And thank you, everybody, for all the questions that are flying in. So this is generally live. The questions are being read off the chat box. So the first question, Darren, are you listening?
Yes. Sorry,
Cool.
I was just being unmuted.
I'm sorry. Question for you, please. Does it damage oxygen transport lung function in humans, particularly asthma sufferers?
Okay. In short, we don't know. There's not enough work really done on the respiratory side of things. There is some interest in using hypochlorous acid for treating the respiratory system at very low concentrations. And there is, I'm aware of those kind of research activities going on. So they've got quite an active program up at the University of Edinburgh where they're using, I think, catatonic salt solution for reducing infections that can be acquired through the respiratory entry points, particularly after cleft surgery. And there is interest in that because of its biocompatibility. But ultimately, the straightforward answer to that is we do not know.
That's part of the consortium to the government in-depth research, isn't it?
Yeah.
Good. So another question, can you use the hypochlorous acid as an irrigation within the aerosol produced by the dental instruments? So you attack the problem at source.
Well, actually, again, there has been previous work, published research using hypochlorous acid or electrochemical activated solutions as an irrigant in the dental sector, particularly for root canals and things like that. It would seem, I think, on first observation, that the use of hypochlorous acid to perhaps or potentially use that to cool the equipment could have a double effect. So you could use it maybe to cool the equipment and possibly to suppress any aerosolization or viral loads. But these things are all sort of interesting concepts. What they really lack at the moment is really good detailed research. So very good ideas. I immediately want to go off to my lab and start working on them, but it's not a straightforward answer that I can give at the moment. But in theory, it's certainly promising. Yes.
Yeah, good. Another question, Murray, one for you really. How does the static fogger ensure all air is decontaminated? So this is the room fogger.
Yeah, it's a good point, and it's one of the, when you say static, what's that being referred to? I presume that the handheld?
No, I believe static being the room. Well, let's assume for the sake of the answer, this is referring to a fogger sitting in the room.
To disinfect the surfaces?
No, disinfect the air.
Sorry, okay. Yeah, well, that's, you can probably answer that better than me, Phil, and I'm probably not the expert on this, but as I think we've said, we need more research to determine the correct parameters. But once we know what that is, we will be able to set a prescribed limit, we hope, for fog concentration or a certain parameter, which will ensure there's evidence backing this up that once you've got the correct amount, we're watching the correct parameter, we can introduce the correct amount of fog into a surgery to decontaminate the air in the surgery, working out the size of the surgery, working out perhaps the humidity or the dew point in the room to ensure all air is decontaminated. But as we've said, we need more information, we need more testing, research done.
Yeah, absolutely. That's good. Right, so Darren, can I use this as a hand sanitizer? It'd be much nicer than using alcohol.
Okay, so again, I mean, the legislation around all of this is just problematic, I'm afraid, at the moment. There are some instruments which are being currently levied, which means you can sort of move away from the commercial products that are currently available and have gone through the certification route. So there is a derogation element to the use of biocides at the minute, and I think they provide a concession for 180 days. I think that's a formalized process. In theory, yes, you can. However, I mean, the way that they test for hand sanitizer actually is interesting. They actually get lots of participants. They get the participants to dip their hands in non-pathogenic E. coli, and then you apply the solution according to manufacturer's instructions. Now, all hand sanitizers have gone through that level and kind of testing. I think, depending on the type of hypochlorous acid solution that you're using, it would be perfectly suited for a hand sanitizer. However, the concentrations and the contact time and the mode of application has to be tied down a little bit. And I think that's, you know, those are the types of questions really that need to be addressed. We have had a lot of experience in using it. It's certainly very gentle on the skin compared to other chlorinated compounds. Certainly much more gentle than alcohol, which is very effective, but nevertheless has a tendency to dry your hands very quickly, as well, of course, as giving you a good shot of alcohol vapor. And that's problematic if you're doing that on a regular basis. So I think, yes, it could. Is it, are there products out there that can be used as hand sanitizers within the current legislative landscape? That I don't think so. Maybe there is, but I'm not sure.
Okay, good, thank you. Can you use the hypochlorous acid made by the 2-3 machine in dental unit water lines? I'll answer that one. The answer is yes, but the actual solution, the mix of water, whether you add a bit per bottle or whether you do it only as like a purge, we're actually working on, but the science behind that has actually been proven many years ago as a good biofilm removal. And obviously we know that the right concentration is completely safe. So yes, it's good for DUWLs, and that applies to the homemade brewers, we're calling it, as well as probably any other solution on the market. But you've got to know the concentration. That's really important. There's quite a few questions flying around as to the required ppm and the required ORP. The answer is you've got to have a blend, and the solution we've got has got a very unique blend of the right sort of parameters. You can reduce one and the other one goes up, and vice versa. So it's not simple answers, I'm afraid, to try and cover those off. What about hypochlorous acid in a low concentration as a mouthwash in pre-dental treatment? Let's have Jonathan on this one. Keep you away, Jonathan, please.
Right. Thanks for that. Again, pretty much like everything we're saying, these are all fantastic thoughts and potential applications. But I was actually bemused when we were working on the FTDP documents that even the concept of using an over-the-counter mouth rinse pre-procedurally was questioned in terms of effectively gaining consent from the patient because of the fact that we are asking them to rinse with a solution, any solution, purely for our own protection and not actually for their benefit, which is a valid point. So I would say at present, until we have the necessary research and evidence to know what concentration and exactly what the parameters should be, I personally would not recommend that anybody do that for the time being.
Yeah, good, thank you. Now there's a suggestion that fogging is to only decontaminate the air and should be used in addition to your routine cleaning of surfaces. Now I'm going to half answer that one. Fogging is hopefully going to be suitable to decontaminate the air, but fogging has been proven as suitable for decontaminating surfaces. So you can spray your surface in a traditional way, and you can fog a surface. The point being is to not let it get really wet and damage it. That's the sort of issue with fogging surfaces. But there's plenty of science around that. You can, can't you, Darren? Air to use a fog to decontaminate a surface. That's proven, isn't it?
Yes, I mean, the crucial difference is the dwell time and the fogging time. So to clean, I mean, to pass the sort of standard tests, you require fallow reduction, and I think 3 or 4 locations in a room. It's based on an old, on a French standard, and it's recently come into force. Obviously, to do that is very different than cleaning the air. So what you cannot do is assume that, you know, if we do, I'm emphasizing Jonathan's point here, if we come up with some best practice guidance for using this particular approach, you cannot assume that that then translates in entirety to cleaning surfaces, because the two are different challenges, and they will require possibly, you know, different concentrations of hypochlorous solution maybe. Certainly, they will require different contact times. So the contact time, and if you think about, you know, if you're static on a wall, yeah, as opposed to bouncing, very tiny particles bouncing around in a room, yeah, and the sort of, you know, the gas laws, if you're static on a wall, you need enough of that fog to touch the surface in a renewed way time and time and time again. So that really becomes a much harder task to do. My advice is to certainly treat all your surfaces that you currently treat as you would under your normal cleaning regime. I know the dental practice has a well-established protocols for that sort of stuff, but by all means, think about fogging in the long run for decontaminating your walls. And that is, I mean, you could, you know, if people have the right certification standard for that, then that is something that you could potentially do very, very soon if they have that information. But you need to get that information from any supplier. And of course, this still would be without human beings in there, and this would be at the end of a day or the end of a week or whatever. But to me, anything stuck on a wall and is stationary, I think that can be dealt with, you know, fairly easily, really. Anything that's moving around and in the way that we're talking about here with arising from AGPs, that's really the focus of the main problem.
Great, thank you.
Bill, can I just come in on that point as well? I would like to make a point just in terms of general infection control practices in a dental surgery, which is, we always have this understanding that you cannot disinfect or even sterilize a dirty surface. So I think cleaning the surface down first with some form of detergent and or cleaner is probably, until we know more, still going to be very, very important. And even when we know more, if the surface is soiled, then whether it's a liquid or a fog, it's not going to get to the microbe. So it's very important that surfaces are clean before we try and disinfect them.
Yeah, thank you, Jonathan. That's bang on. The soiling has to be physically removed. Thank you. What's the difference between H++ and DentiGuard? It would be the ORP, it would be the ppm. And if you look on the bottle, look at the dwell time that's been referred to, or the kill time. It would probably say under a minute. Now, under a minute in the air isn't going to be very effective. We're looking at sub 10, sub 2 seconds. Okay. I thought this one was going to pop up. Sterilox has been used for many years in dental water lines. Can we use HOCl in the same way? The answer is yes, it's all the same. Excuse me, running through. Does the hypochlorous acid damage uniforms when spraying lingers? Yeah, it's still really, really good questions. As Murray touched on, it's about the amount we apply and then the removal of it. Marlies, if you use a fogger, start on one end and go to the other end, it means all surfaces are being decontaminated. And then after 10 to 20 minutes, as per guidelines, we can perhaps get going again. Yeah, Jonathan, you touched on this, didn't you? And it's been really through the whole webinar, is the human element is something that we feel won't be suitable for part of an actual standard. I think we can all agree there may be some effect by doing that, but we need absolute science on it and controlled, measured amounts of a solution applied, and the same of removal. And if Jonathan, you want to add anything to that on the handheld?
I agree with that completely. I think that it's probably, first of all, we have enough to do after procedure. So to utilize one of your staff to actually stand there fogging your room probably isn't the best idea from a time management standpoint. But second of all, as you say, the human element is critical. And we need to come up with a solution that effectively is a push a button, close the door and leave the room solution. And then we know that it's automated. And as was mentioned earlier, if it's the right room size and the right amount of fog for that room size and the right conditions in the room, we know the job is done. So I think those parameters have to be adhered to without fail every time if you're going to rely on that air being clean.
Thank you. All right, questions are still coming. A question on if a fog is to be manually operated, there's specific on minimum PPE. The answer to that question is we're not advocating manual fogging at all. There's too many unknowns, too much human element. We've had reports of people feeling giddy, even fogging under a FFP3 mask. It may be carbon dioxide build-up. We don't know. So I hope that answers that question. So are we saying fogging is, at the moment, is useless? Who wants to answer that one?
I'm happy to answer it because I actually answered it in what I said earlier. It's not useless at all. It has fantastic, huge promise. And as we said, it's been proven in several other industries. And there's no reason to think that we won't find the right setup in dentistry. But what's important is I'm not aware of anything to say that we have the right setup yet. And we certainly don't have the research to prove that we have the right setup. And that's what we need for our safety, for our patient safety.
Yeah, thank you. Murray, we've been asked what time frame we're looking to get the fogging machine ready for market.
I wish we knew. We've been spending hours and hours on this. We would love to have had it ready for today, but it's not just the machine. The machine is important and we know what the machine's got to be, but it's evidence, the parameters, as Jonathan's mentioned. We need to know what the machine needs to do to be able to it's a whole process to put together. It's the fogging and then it's the removal of it, because that's important, and the machine's got to be able to hit the parameters needed to ensure efficacy. So we'd love to be able to put a time frame on it. We're working very hard. We have a prototype which meets most of the criteria, but we still don't yet know the parameters, and I think that's the really key part which Darren's research will help us determine as soon as possible. But we will certainly keep. The key thing with the biofog is to keep everyone updated. This is exciting. This has huge potential. We are doing all we can to keep the industry updated.
Yeah, thank you, Murray. We're pretty confident we'll get something that will have as much science as you've heard tonight behind it, but the actual rest of it may catch up with it later. So as a takeaway, can we be confident we're going to get a solution for rapid disinfection? The answer is yes. Someone's asking about the fallow time statement. Jonathan mentioned this earlier. We believe it's been delayed and likely to come out tomorrow. That's James Meta asked that question. I think that's okay. Dr. Shah, if I'm correct, you're saying we need to wait 1 hour fallow time before we can wipe down the surgery. It needs to be disinfected first, then use the fogger. So basically has no effect on fallow time. There's quite a few bits inside that. Did you catch that, Jonathan?
Yeah, I think it sounds like it's going to be a long, arduous process is what that person is suggesting. But in actual fact, the recommendation would probably be, I stress, that you might do an initial cleanup of your instruments and take them out of the room as you're doing now, and then leave the room to be fogged and come back and as a secondary clean, wipe down all the surfaces, because besides fogging, you also want to give an opportunity for whatever is in there to continue landing on the surfaces. So you come back and you would give them another clean. So you could do an initial wipe, fog the room, which for all we know, we hope might be a 5-minute process or a 10-minute process. We don't know, if your research shows. And then it's just a matter of a secondary clean. So I think, you still might be looking at a 15-minute process, but that's a far cry from a 60-minute process. So I think there's still some potential benefit there.
Yeah, I totally agree. Another question, would there be any issues using hypochlorous acid in conjunction with hepatite filtration and cleaning? I'll try and answer that one. The air purifiers that have been sold, as long as you get the correct purifier with the correct filtration levels or kill properties at the correct flow rates for the right size room, they can be effective, definitely as a background cleaning. But what we're trying to produce here is something that's even more rapid decontaminant, not just of the air, but also the surfaces that have already been physically cleaned. So what we're talking about is a real gold-plated approach to every single possible issue within your dental practice after an AGP. Darren, do you want to round us up with anything? You've been quiet for a few minutes.
Yes. Yeah. I mean, I think the prospects are good. I don't think. We have to be realistic. I wish I had more answers, definite answers, but we're just in that space which requires some really great sort of science. I mean, this is not sort of something which the UK's been lagging behind. It's just really, really difficult science to do. We've submitted an expression of interest, more or less, to the UK government to try and perform an accelerated research programme, which at the very least can demonstrate efficacy within that airborne space. Not efficacy on a fixed load on a wall, but efficacy in mechanical air, on all those airflows and all those complicated things. If we can show that, then that's going to be a good bit of evidence, which I think pushes this in the right direction. So there's going to have to be a little bit of pragmatism, I think, and I'm sure that, maybe I'm not sure, maybe you're sure, that the particular dental sector will perhaps organise what a practical and pragmatic way is. But the most immediate thing really is dealing with AGPs. and the risk that that can have on the potential transmission of COVID-19. Because actually we do need our dental spaces open, we do need our clinical spaces open, we need to access healthcare at many, many levels. And this is not something that is unique to dentistry. This also affects many other clinical spaces. And I'm hopeful that the government will either have come up with a solution already, or if not, at least give us the opportunity to help provide you with some solutions. That's all as a scientist we can do, is provide you with a technical, evidence-based, solution to some of the problems. There's no magic bullet, but hopefully we can at least supply some solutions that address those really, really key challenges that a lot of you guys that have been here tonight listening to the webinar clearly face on a regular daily basis. And that's my motivation. It's not, it's not sort of selling fogging equipment or solutions. My motivation is purely knowledge based. And I don't, in order to understand the problems, I need help from guys like you to inform me as to, what that problem is, because I need to work in partnership and in collaboration with you guys, really to try and come up with some reasonable solutions. And there's lots of hurdles. There's safety, there's regulation, there's technical specifications, there's equipment, there's fogging, there's people, there's fallow time, and there's doing all that. And of course, everything on all levels, those things need to have been done yesterday. But actually what we've got to do is to affect what's ahead of us. And that's what we need really, is lots of enthusiasm within the sector to try and address some of these big problems. And I think the UK science base has a key role to play in that.
Yeah, thank you, Darren. Thank you, Jonathan. Thank you, Murray, Fraser, and thank you everybody that's joined the webinar. Trust it's been interesting. Trust it's been useful. We've certainly enjoyed your questions. We're going to be recording this. It's going to be out live, and so you can watch it again. The questions that have come through are still more learning for us on there. And as Darren says, we need your input. So please don't disappear on us. Please keep connected to Anglian, to Biofog UK, and any ideas, any questions, you're really, really part of this really exciting push forwards. So thank you, everyone. Have a lovely evening and good night.